Hi everybody, so in this short segment we're going to talk about what happens when DNA is altered and what happens to the resulting protein product you get when changes are made to the DNA code. So we'll look at the effects mutations have on protein synthesis. First off, when you have a change to the actual genetic code, you're going to ultimately get a change in protein structure most times, because that makes sense, right? If we go back to the idea of when we watched transcription and translation, in the whole process, you had the gene became the transcript then fed that message through the RNA, and voila, you had your polypeptide chain. So if I alter the DNA, that could alter the transcript, which then could alter the actual outcome that I have. So that's a mutation by definition, an actual change to the genetic code. Now, most mutations are deleterious, okay, which means they're not good, okay? They usually cause bad things to happen. Because as we know, you change a protein's primary structure, you could change its shape, which then changes its function. And if we're talking about important enzymes, if we're talking about key proteins, that could be a really big deal. So a few types of mutations. We have a base pair substitution, in which case you get a different letter, okay, than you would normally, okay? So base pair substitution. So for example, here's the original. If we get a base pair substitution, notice here that on the gene, it's a T. Well, the substitution piece is that instead of a T, I have an A. Okay. Now, when that actually gets transcribed into the codon, that codon, okay, then codes for valine instead of glutamate. Okay, and this is one of the things that happens in sickle cell anemia. We can also have a second type, which is an insertion or a deletion frame shift. So instead of just replacing the base, which doesn't alter anything on either side of it as far as the actual code, any of the other ones, it only alters that one amino acid. An insertion, like actually adding in a base or removing one out, totally shifts your threes. So instead of reading it, AAC, if I were to take that A out, okay, that first A, so if I were to have AAC, right, let's put it over here. So AAC, let's say, and let's say the next one was a T after it. Well, if this gets removed, instead of my codon being AAC, now my codon is ACT, okay, which not for nothing, you'll actually code for a stop if you were to do that. So here's what a frame shift deletion looks like. So here's, again, that original, right? Here's my original, CTC. Well, I've removed, this T has been removed. So instead of going CTC, which codes for glutamate, now I get CCC, which then codes for glycine, and everything subsequently after that frame shift has now been changed. Okay, kind of a big deal. So what could be some causes of mutations? Well, there's a lot of things that could cause it, okay? We can look at rates. The thing about mutations, though, is a lot of times they're random, and to be able to calculate the rates of mutations can be very difficult, but given certain variables, we can get an estimate of how often these occur. So the big thing is that, remember, this is occurring in the DNA. So what's going to cause the change? Well, one could be when the DNA has to replicate itself. So as the cell's getting ready to go through mitosis and there's a replication of the DNA that occurs, or even meiosis for that matter, errors occur as the DNA is replicating. I mean, there's built-in, error proofing, but sometimes mistakes get made. Those are pretty low key. So this can be caused by the repair genes being inactivated and not working right to go through and proofread. Could be a number of reasons, or the proofreaders actually just simply miss something. Okay, environmental factors are a huge cause for mutations. So things, and we call them mutagens, anything that can cause a change to the DNA. So things like radiation is a known mutagen. It will alter DNA. This was one of the ways that Beadle and Tatum found out that genes code for a protein product, was through exposing them to radiation type things. So that's why you're only limited to a certain amount of x-rays a year, and why they try to localize that exposure so you're not getting full-on x-rayed across the board. Exposure to radiation and things like that, where is one of the things that led to the cancer that Rosalind Franklin got. Chemicals are also a huge factor in causing mutations. So you've got different environmental chemicals, chemicals in what we eat, if you smoke, all these things that can cause actual mutations to occur. Now there's differences. If I have a mutation occur in one of my somatic cells versus an actual germ cell, obviously the outcome can be very different. In a somatic cell, I might have that one cell get exposed, but the rest of my 10 trillion cells might be okay. All right, so maybe it's not the biggest deal if one of my somatic cells. Now, if it's a germ cell, though, that's been exposed, every resulting cell after that for the organism is going to have that mistake. Okay, so the consequences are different depending on what it is. So just a couple things, and this is actually what I'm looking at here, is a shift that occurs for sickle cell anemia. Okay, and it's not the full-on protein being changed, but simply a single amino acid shift that occurs. And that's what causes the misshapen hemoglobin, the inability for it to attach to oxygen well, and you wind up with the cells that, remember, when they squish through the capillaries, stick, and then they can't do anything from that. So yeah, these changes have consequences, unless of course the change happens at a codon that codes for the same protein. Because remember, not every codon is codes for a unique amino acid. We have some that actually code for multiple, have multiple codons coding for that same amino acid. So if the change occurs there, the resulting protein will be the same because you coded for the same amino acid anyway. There you have it, little mutation look, and we'll go back and compare that to what you saw in the protein synthesis investigation, when you actually saw how one of those deletion mutations actually affected the outcome of your protein. So take it easy, and we'll catch you guys later.